afternoon, class. So, in today's botany lecture, we are going to see the reproduction pattern inside the Pteridophytes plant. So, as we know, the Pteridophytes, also known as the vascular cryptogams, and we have to see how they can reproduce. And the reproduction is a fundamental process that every living being has to be go through. Either it can be the animal, a little microorganism, plants, fungi, alga, whatever the thing which has been have a life or which is in the living environment, that has to be and they should be go under the process of reproduction for their continuation process. So first begin with the introduction part, and in this we are going to see how and what kind of characteristics present inside the pteridophyte plants. After that, we will going to see how they can reproduce. So the pteridophytes are the first terrestrial vascular plant. Okay, so if we start with the right in the form of evaluation, or you can say second term is evaluation, first term, and second term is evolution. So actually, evolution is a different thing. That is a chain of series over a time period. What in the case of evaluation? So actually, when a thing is evolving, so characters are evolving in a manner of adapting more. So basically, when we see in the classification part inside the plant, we divided them into the two major groups, that are the cryptogams and phanerogams. So in the cryptogam, we consist and we put the plant which have a low tendency, or we can say they did not evolve as much as the phanerogams. So the cryptogams, they do not have seed even inside them. They do not have flower. They do not have fruit. So these are the characters which are lacking inside the cryptogram. And also a very important thing that, or you can say a very important compositing unit inside the plant, that are the vascular plant. So these pteridophyte, among all the cryptogam, that the thallophytes, bryophytes, and pteridophytes, only pteridophyte have this slight luxury, I can say, to being a terrestrial vascular plant. Why I used the word slight? Actually, the all the cells or all the part of this vascular tissues. Actually, vascular tissue are possessed, or they can be as called like the xylem and phloem. For water conduction, we have xylem inside in the plant, and for the food transportation, we have the fluid. But all the cells which has to be present inside the xylem and phloem, they are not present in the pteridophyte. So they are the one which just take initiative, or they are just kind of primitive in the case of having the vascular bundle inside them. So very fundamental kind of the cells of vascular tissue are present inside the pteridophyte. There is no very developed kind of vascular bundle are present in pteridophyte. Carlos Lianas, the one legendary modern father of modern botany, classified them under the group Cryptogame, and Pteridophyte occupies a transitional position between the Bryophyta and Spermatophyta. So, according to the Lianas, he basically put these Pteridophyte in a transition position in between the Bryophyte. Bryophyte one, starting with the plant classification, we start with the Thallophyta. After Thallophyta, we having the Bryophyta. After Bryophyta, we got it the Pteridophytes, and this is a linkage between having the seed forming plants like the gymnosperm. So they can be in between the transition position in between the gymnosperm and Bryophyte. So they have some characters. Have some character who can put them into the phanerogams, but not all majorly character. Or we already talked about it. Similarity degree and differences degree. Similarity degree always has to be higher to put them into a group. So that's why the character similarity of the pteridophyte, they have higher as linked to the bryophyte, not as linked in the spermatophyte. So that's why we put them into the cryptogamy, not into the phanerogamy. They do not produce flower and seed, so they do not have this luxury to produce flowers and seed. Hence, they are called a cryptogram. So that's why they can be called as cryptogram, but they reproduce. Okay, so this term cryptogram actually this is this why we are calling them cryptogram, because this Greek word has a meaning. Like in the Greek, we have meaning of the secret marriage, but the secret marriage in the sense of the plant reproduction, that their reproduction pattern cannot be seen like we can see them inside the angiosperm in the form of flower. When a plant get a flower, so we can be easily identified that they are basically come into their reproductive phase. But inside the pteridophyte or bryophyte, we cannot be able to analyze by just seeing or by just look at the plant to tell them that even is are they in their vegetative phase or are they in their reproductive phase. More than 12,000 species of pteridophyte are found on Earth, so there are a wide range, 12,000 species, and they are having this patron meaning feather and phyton meaning plants. So, term this patron meaning, that is basically for their feather. Feathery appearance, I will show you the diagram so you will be clear why we are calling them the feather. So now you can see the diagram here. So by seeing the diagram, they look like a feather. So that's why we call them the feather plants. And this is a whole reproductive cycle. This is a whole life cycle which we are going to study. But we will take a diagram in after. But you can see that how many phases we needed to study in along to studying about how pteridophytes can go under the reproduction path. So reproduction begins with the sporangia formation. So what is sporangia? So as a name suggesting, this is a unit which going to bear spores inside. So the sporophytic plant reproduce by means of a spore produced in the sporangia. Their reproduction, you can just say the another name for their reproduction is sporangiogenesis. Means producing spores inside the sporangia, and reproduction will take place by these spores. So now we will take a look how the sporangia of pteridophyte look like, what are their characteristics, how they can be differentiated with the others. So the position, if we see that at what place they are present. So the position of sporangia may vary in different groups. That can be like we have a lot of groups inside of pteridophyte also, because pteridophyte is a bigger unit. It is a subdivision. We have orders. We have genus inside them, lots of. So they may be born on the stem. So this is sporangia, this reproductive unit, they can be born on the stems. And when they are born on the stem, there is a term that can be called as called pollen. In the Xylotem and Rhinia, Rhinia is even a fossil plant, but still they belongs to the pteridophyte. Or on the ventral surface means adaxial surface, opposite to the abaxial surface. Surface of the leaf, if we it is present on the ventral surface, we call them the adaxial. Same meaning, ventral and axial, they have the same meaning. Surface of the leaf, we will going to call them the foliar sporangia, and it is present inside a lycopodium and selaginella. Or even they can be present on the exile of the leaves, that is called ophioglossum, and they are very beautiful when we look them. I will show you how when they are present on the exile of leaves. You can see how beautifully they present on the exile of leaves. They look very charming when we see them, and even at that time we can even count how many sporangias are present, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Majorly lower leaf, they have lower amount of sporangia. As they begin to the topper portion, they will increasing the number of the sporangia. It also depend, and even most of the time, mediocre leaves, the middle leaves, they have more number of the sporangia as comparison to the lower. And upper surface do not have that much surface area actually. Upper point axis of the leaf, they do not have much surface area, so that's how many sporangia are going to put them inside. So, okay. These are the kind of sporangia that can be seen inside the pteridophyte. So the sporangia contain leaves are called sporophyll. The leaves I just show you on which sporangias are present. They have a specific name here and they are called the sporophylls. So the sporophylls may be scattered. They can be present scattered. They do not like to present in a uniformity like one by one or alternative in fashion, parallel in fashion. They just present whatever they like to be distributed. Sometimes uniformly distributed, sometimes they know their manners. So in terrace and adenitum and other forms, they can be in the uniformly, or sometimes they can be grouped, or sometimes they like to be grouped and different areas to form stabili, that is in the Selaginana and Ecosystem. So sporophyll can be three types: scattered, like Ogodium and Selago; uniformly distributed in the case of Taris and Adenitum; and grouped in the form and definite area to form stabili, then Selaginana and Ecosystem. In more aquatic pteridophytes, the sporangia are present within a specialized structure called sporocarp. Yes, like a normal plant, normal terrestrial plant which lives on the earth, they do not need a special covering to protect them from water, excessive water actually. So that's why an aquatic pteridophyte, which the pteridophyte which lives inside the water, they need some special kind of covering. They need some special kind of safety for their sporangia. At that time they have this structure called sporocarp. An example for these structures are Zola, Salvinia, and Marsilia. They are the aquatic pteridophytes. So, on the basis of mode of development, the sporangia are the two types. Okay, mode of development means how they are going to develop from a very starting age to their development, how they are going to develop. So, this two kind of development is eusporangiate and the leptosporangiate. Now we will going to see what are the difference between eusporangiate and leptosporangiate. So in the eusporangiate, diplosporangia develop from several sporangial initial. Okay, like there are several of groups. There are several of cell 1, 2, 3, 4, 5, 6. They come together and make a sporangia. Okay, that will called the eusporangia, and this happen inside the salutum, lactopodium, and selaginella, where a group of cell come together and make a sporangia that is called the eusporangia. This you can say the most of the cell in here they are going to come at their places and going to make a sporangia that can be called as eusporangia. And these are the leaves on which these sporangias are bear. They are called sporophylls. They are bearing sporangia. This orangish features, spherical in shape mostly, containing four megaspores. This is a megasporangium. This is a microsporangium. So I think you can clearly see the difference between the megasporangium and microsporangium by even in the color. Color is different. Megasporangium have yellowish texture and microsporangium, they are the male sporangium. They are having orangish and broad. They are broad and they are multiple. They are not a singular unit like the megasporangium. They are containing four megaspore here. One, two, three, four. Four units there. Microsporangia, they will be with many microspores. They do not have a definite number like we are going to be two, four. They are like millions. So they can be present inside the sporophylls. Most of the time, megaspore laterally present, and they present on the central axis. Microspores or microsporangia, they present on the central axis, and megasporangia present in a sideward manner. And yeah, I didn't tell you about leptosporangia. Leptosporangia. Eusporangia will be formed by grouping of a cell. Leptosporangia develop from a single cell. Okay, one single cell going to make whole sporangia. That is called leptosporangia. And when the group of a cell is going to form a sporangia, that will be called as eusporangia. Now, on the basis of maturity of the sporangia, the sori are the three types. So, this sori will be called as a single unit. Like this is the sporangium, and inside this there will be a sori that is a single unit that will be called as sorry. The multiple unit is called as Surai, and the single unit is called as Saurus. Simple Saurus, if we see a Saurus in which all the sporangia originate, grow, and mature at the same time. Okay, simple Saurus when we are talking about the simple Saurus, so in which all the thing like all the sporangia within it, they will originate at the same time, they will grow at the same time, and they will going to mature at the same time. When all the things happen at the same time, they are like the simplest thing, so that's why we calling them the simple suras. The form showing such conditions are grouped together as simples. So the form everything at every condition grouping together, this is are the characteristic of having normal. This is a beautiful diagram. Graduate Saurus, that is second one, second kind of Sora here. Here sporangia develop over a time period. Okay, there a definite time period has been required for sporangia development. Now, what happened? How they were divergent with each other? Central part of the sorus has mature sporangia, and the peripheral part has younger sporangia. They do not mature at the same time. Central part have maturity, but the end parts, the peripheral part, they do not get mature at the time of central part mature. So they remain younger, or you can say they develop later even. So this kind of situation seen in the Marsilia, Scythia, Hymenophyllum, and they forms showing the condition is called gradate. So this condition when the central position got mature early and the peripheral portion do not get mature at the same time, this kind of situation is called as a gradate. And the sorus which are having this kind of situation in them, they are called as gradate sorus. Mixed sorus. Here, the mature and immature sporangia of different ages are arranged in an irregular fashion. At the gradate spore, you can identify that what kind of spore is going to mature at what time. Means if there's a maturity and another one that didn't mature, you can identify at what place they have. But in this mature, immature sporangia of different ages, you cannot identify them. They do not show any kind of pattern like center have maturity, peripheral have immaturity. They just, mixed up. They just have all the thing immature and mature sporangia just mixed in between them. So that's why simply we call them the mixed sorus. We don't even want you to get so much confused. So just we tell them the mixed sorus. Now we are talking, these were all about the sporangia. Now we are going to talk about the spores. They are the unit which going to make plant reproduce. Spores. They firstly their division. How spore forms. Meiotic reduction division of spore mother cell produce numerous haploid spores inside the sporangia. Okay, now we have sporangia. In a sporangia, we have spore mother cell. In spore mother cell, there will be going to the meiosis. Meiosis will be happening there. After meiosis, they will be going to produce some daughter things. Whenever a division happens, daughter cells will be formed. Now the daughter cell which are going to form by the spore mother cell, of course, if it is a mother of a spore, they are going to produce a spore. And these spore are has to be haploid because they come from the meiotic division. So that's why they come from a meiotic haploid cells will be formed, haploid spores will be formed. If all the spores produced are of the equal size and shape, then the plant is called Homosporous. Now, all the spores which has been produced by the spore mother cell, if they have the equal size, equal shape, everything is equal. So they will be going to call that plant the homosporous plant. Means their all spores are the same kind. So it can be seen the Lycopodium, Equisetum, Dryoperis. We can see that. And if they are of the two different size and shape, then the plant will be going to called as a heterosporous. If the condition is opposite to the homosporous, then we are going to call them the heterosporous, and they can be seen in Selaginella and Marsilia. In the heterosporous type, the two different type of spores are produced in separate sporangia. So there's a condition in the heterosporous life cycle. Now we have to produce different kind of spore in different sporangia also. We cannot produce both the kind of spore inside of one sporangia. It has to be produced in a different sporangia. Like the smaller spores here, the smaller spore inside the pteridophyte, they will be calling themselves, or we will be calling themselves microspores. Microspores, and they are called as male spore, and they are going to produce in the microsporangia. So, what the three things you have to remember? Heterosporous means different kind of spores. If different kind of spore, they are going to produce in different kind of sporangia. In different kind of sporangia, there will be one for male and one for female. For male, we are having microsporangia, which are going to produce microspores, which are small. Okay, they are small, they are male, and they are microspores. Second thing that is called megaspore. We will see it later. Megasporangia, they are called as the female sporangia, or you can say the spores which are going to form inside it. They are called megaspores. They are larger as comparison to the microspore. Megaspore are larger. They develop into the megasporangium, and they are the name for the female unit of the plant. So, microspore after germination produce male gametophyte. When germination happen in the microspore, they are going to produce male plant. Okay, male gametophyte, not plant. Sorry, male gametophyte. When the germination will be happen in the megaspore, they are going to produce a female gametophyte. So I think condition is clear about the heterosporous life cycle, megaspore and microspore. Microspore develop microsporangium, megaspore develop megasporangium. Mega we are talking about female, micro we are talking about male. Male is small, female is large. So now, next in the sporophyte with megasporangia are called megasporophytes, while sporophytes with microsporangia are called microsporophytes. So, like the sporophyte, the fertile leaves, they has to be associated with these microsporangia because if they are not present, they cannot make us. So that's why the leaves or the sporophylls, which are related or which are present inside the megasporangia, they are called megasporophyll. And the other leaves, which are associated with the microsporangia, they are called the microsporophyll. Next, we are going to begin. This was all about till now, whatever you have been studied, okay? So this was all about the sporophytic stage, but now we have spores and meiosis has been happened inside the plant. We are going towards the gametophytic stage. Reproduction is divided into two stages, okay? Sporophytic stage and gametophytic stage. We just completed the sporophytic stage. Now we are going towards the gametophytic stage where everything is haploid. The spores germinate to form haploid, gametophyte, or prothalite. So the first thing, when these haploid spores going to germinate, they are going to make the haploid gametophyte, or another term for them is called prothalite. The gametophytes of pteridophyte are small, inconspicuous as compared to the sporophyte. So the gametophyte which is going to develop here, it is very small if we compare them to the sporophyte because they do not live longer. They do not have to live longer. They just are present for a certain time to go for the fusion. The germination of a spore in the homospore form of three time bipolar, tripolar, and amorphous or irregular. So they can be three type here. The germination of the spore. Bipolar, two will be on two direction, tripolar, three direction, irregular, any direction. So the gametophytes are of the two type in homosporous form, and the development of gametophyte is exoporic, in which the prothallus develop outside the spore wall. Okay, like in this case, when they are germinating or when they are making the gametophytic stage, then the prothallus, which is developed, if that is developed towards the outer side of the spore wall, what we are going to call them? Exosporic. Exo means outer, And outer for what? Outer from what? Spore. So we can call them the zoosporic. Therefore, it is vulnerable to outside environment gametophyte that develop from the heterosporous are endosporic, in which the development of prothallus is confirmed within the spore wall. The thing which is newly formed, they are very sensitive to how they can going to live in such a harsh outer environment. So that's why the gametophytic stage that has to be developed by the heterospores, they have some endosporous development or endosporic development inside the wall. Spore. But homospores are like they are like strong ones. They can survive anything because they already come from the same background and they know how to deal with everything. Heterosporous, they feel like we are some kind of weird, so that's why they think good to germinate inside. The development of endosporic emetophyte is independent to external environment. So they depend on the plant condition, whatever they want. It's their life. If they wanted to go outside, that we just have to remember their movement name. If they are going outer side, we just remember as a exosporic. If they are developing inside the spore wall, we are going to remember as a endosporic. Next, the sex organ. So they're going to have two sex organ as everyone have. So the gametophyte or prothalli, singular prothallus, okay, prothalli, multiple term. If we're talking about the singular prothalli, we call them the prothallus. Here the sex organ, male antheridia. Male organ is here called as antheridia. Singular antheridium. Female archiconia, multiple term, singular archiconium. Nium is a suffix for the singular. And nia is a ia, it's basically ia suffix that is for the plural inside the this whole reproductive organ of plant. So these six organs are embedded and uh, impregnated in the prothallus, so or are protected. So they can be inside the prothallus, so they can be just have some in their depression. So we will be going to see each one of these, okay, antheridium here. So the antheridium is a sessile or short-stalk globular structure surrounded by a well-defined jacket inside containing androzoid or anthrozoid's mother cell. So as an antheridium, so it is a male part, and firstly it is sessile and short. Sessile means they do not have any kind of stalk formation. Sometimes they have some short stalk or globular structure. Basically, when we see their structure like globular, sessile is no attached directly attached to the substrate. Surrounded by a well-defined jacket, they have this everything need a protection. So they are the very sensitive things, so they need protection. So that's why they have this well-defined jacket. And in this jacket, they are having this endocytes and anthrozoids mother cell. Now, this anthrozoid mother cell, what is he going to do here? So each androcyte give rise to a single motile anthrozoid. Now, what's their function? They are going to give the birth to the single motile anthrozoid. The number of anthrozoids per antheridium varies from 4 to few thousands. They can be 4, but they can be few thousand. They are unpredictable. You cannot predict to them to have like 20, 30, 40. They can be whoever the condition they are present in. If they have everything is in favorable condition, they can produce as much as male can they can produce inside. But you have to remember in all this slide that antheridium is sessile and shortly stalked. Inside them, we have anthrozoid mother cell. And what anthrozoid mother cell is going to do? Anthrozoid mother cell going to produce anthrozoids, and anthrozoids is a single and motile. Motile means they can move. And how many numbers they can be have inside from 4 to thousands. The anthrozoids, if we talk about a now singular anthrozoid which has been produced by the anthrozoid mother cell, so these are unicellular, obviously. Uninucleate, one single nucleus. And spirally coiled bearing apical flagella. The flagella which help them in their motility character. that is spirally coiled, and sometimes they can be multiflagellate. Multiflagellate means multiple flagella can be present, and sometimes they can be just having one single apical flagella. That depend in what species they are going to take birth. Okay, so if they are having a Lycopodium and Selaginella, they will be having just apical flagella. If they are going to born inside the xylotum, isotes, equisetum, fern, they are going to have multiple flagella. So that basically depend in what kind of species they are going to present. Archiconium, the female part. So the archiconium is a flask-shaped structure. Okay, they were the globular structure. They are the flask-shaped structure, consisting of a basal, swollen ventral, and a short neck. So they are having these three part: basal, bottom part, swollen ventral, middle part, and a short neck at the bone. The venter is embedded in the prothallus while the neck is projected. So this ventral part, the upper part, swollen venter part, is embedded in the prothallus while the neck is projected. The venter encloses an egg and a ventral canal cell. Now, what are the things inside the venter? Two things. One egg cell, that is the major issue here, and a ventral canal cell. They are just present here to give the protection and nutrition to the egg cell. Neck is made up of the vertical rows of neck canal cells and inside. These neck canal cells are present here just to make the passage when the anthrozoid come to here, and they just going to disintegrate, just, or you can say that they just going to be degenerated when the sperm come here or the anthrozoid come here, or they make a path for the anthrozoid to go to the egg cell for the fertilization process. So what you have to remember in Archiconium, they are flask-shaped structure. They have three parts: basal, one bottom basal part, swollen venter, and short neck. What they have inside the venter: one egg cell and venter canal cell. Why venter canal cells are there? They are providing them the nutrition and support. After that, the neck canal cell inside them, they are present here just to make the path for when the anthrozoids come there, they're just making the path to come inside. Now the fertilization. So the disintegration of the neck canal cell, like I said, also produce a mucilaginous substance which contain organic compound like malic and fumaric acid. So when these poor neck canal cells get disintegrated just to make a path for the anthrozoids, also at that time by crying they produce two things, okay, two organic compounds. One is a malic and second is a fumaric acid. So these malic and fumaric acid, they basically work, or they basically work a chemotaxic activity to attract the anthrozoid or sperm into the neck canal, into the neck portion. So what is essential for the fertilization? Water needed to be there, because if water will not there, how can sperm going to swim? So, water is essential. Sperm swim over a thin film of water and attracted chemotactically towards the archegony. So they will be like getting influence from this, acid, malic and fumaric. So on that way, they will going to make their path. So the anthrozoids and egg or haploid chromosomes number fuse to form a diploid germ. So we know that egg also haploid number, and this anthrozoid which is coming here, that is also haploid. So both haploid is going to fuse and make a diploid zygote, which is the mother cell of the sporophytic generation. And from this part, and still after the spore, from the spore, we are studying the plant in the gametophytic generation. Now this again plant going to the sporophytic generation when this zygote form. So zygote is the first cell towards the sporophytic generation of the plant again. So now we will going to see the embryo that has to be formed by the zygote formation. So repetitive divisions inside the zygote led to the formation of embryo. And after this embryo, they will be going to form or differentiated into the root, stem, and leaf. So basically, on the basis of their polarity, the embryo may be categorized into two groups. exoscopic embryo and endoscopic embryo. So basically, in exoscopic embryo, the shoot forming apical cell is directed towards the neck of the article, or you can say directed outwards. So the neck, the shoot forming neck towards the apical cell, or you can say the shoot forming apical cell, they will be towards the outer side. In opposite to the endoscopic development, shoot forming apical pole, the thing which is going to make the apical pole, the plumal, but they are directed towards the downside. That is called the endoscopic embryo, towards the base of the archegonium. So if they are growing towards the neck, towards the outer side, they will be called as the exoscopic embryo. If they are towards the inner side, they will be going to called as the endoscopic, bottom to the archegonium. So the basis life cycle pattern of pteridophytes shows a regular heteromorphic alteration of generation between gametophyte phase and a sporophytic phase. Now, the thing which we seen in this whole reaction, or you can say whole life cycle, basically actual, the first thing what we seen the sporophytic phase of the plant when they are making the sporangia. Sporangia, the thing we seen that was a sporophytic stage. When inside the sporangia, in the spore mother cell, when the meiosis happen, spore form, and this is the first stage of the gametophytic generation. So sporophytic, then gametophytic. Now again, when this zygote forms, just this is again for the sporophytic condition. So whole this cycle just move on and move on for the counterparts. Like first sporophytic, gametophytic, again sporophytic, then gametophytic, sporophytic. So this is a cycle. And this cycle or this process is called heteromorphic alternation of generation. And this is the key of the reproduction in pteridophyte. If someone knew everything and you just tell them, like he asked, did you know what kind of life cycle pattern is present inside the pteridophyte? And you just tell them, that person, yes, heteromorphic alternation of generation is present inside the pteridophyte. He know that you know. Okay. What is exactly the meaning of alternation of generation? That these both the generation alternatively present with each other. In this, gametophyte is a sexual and sporophyte is a asexual. Gametophyte is haploid, sporophyte is diploid. The things you have to remember. Okay, so the main plant body is sporophyte. The main plant body or dominant plant body which take over the whole life cycle, that is the sporophyte. And gametophyte generation just bearing for just for the bearing of male and female sex organ and 3D and archegonia for a very short time. They do not live there for a very long. Androzoid male gamete produce in large number of motile, while the egg female gamete are non-motile and are born single in the archegonia. So inside the gametophyte, anthrozoids are the motile one and very large in the quantity, but archegonia, they just born singly and they do not even move. They are just non-motile. Fusion between an egg and the anthrozoid result in the formation of diploid condition, or you can say the zygote. Zygote divide mitotically division into the sporophyte, and the sporophyte plant develop sporangia, which produce haploid spores through reductional division. So this all thing that is written here, I just explained to you. In homosporous members, spore germinate to produce monoecious, homothallic gametophyte bearing both male and female sex organs. This was what's left. We just see how the heterosporous life cycle works. In Homosporus, both the sperm, both the gametophyte, both the gametes, male and female gametes, they will not going to produce by microgametophyte, gamete or the megagamete. They are just be produced by the same kind of spore. Same kind of spore is going to form the male gamete and female gamete. In this case, sex determination take place at the time of formation of antheridium and archegonium. When they are going to make their antheridium on the basis of their spores, then only we can identify them. Before them, when they are in the spore phase, we cannot identify them on the basis of their spores because they produced by the same kind of spores. So this is the life cycle of Homosporous pteridophytes. Okay, this is the Sporophyte stage. Here everything just began, and Sporophyte stage. They are going to form. They are having meiosis inside them. Meiosis is a reduction division, and they are going to produce spores. These spores are similar in the case of Homosporous. They just may cover, they go to into the gametophytic stage, and these two same जो spores हैं, these same spores, they will be going to behave like the anthridium in case of the male gamete and archiconium. Then only you can realize what spore is, what purpose for. After this haploid condition, they are going under the syngamy process, or you can say fusion, fertilization process. And after attaining the fertilization process, what they are going to form? They are going to form a zygotic condition, that is a diploid cell. And plant again come into the sporophyte stage. So the sporophyte, gametophyte, sporophyte, gametophyte. This is called alternation of generation in a homosporous dendrophyte. In heterosporous, micro and megaspores on germination produce male and female gametophyte respectively, both. Here the sex determination take place much earlier, so we can easily identify the what kind of spore is present just by seeing them, because I told you microspore is a small one and megaspore is a large one. So just by seeing the spore you can identify them. Here differentiation can take place only this part. Sporophyte, they are going to have meiosis, reduction division, and going to make a special kind of spore. Small one, microspore, male. Large one, megaspore, female. They are going to make their own gametophyte here. Okay, male microgametophyte and megagametophyte. Then going to produce Androzoids and Archiconium, and they also going under the process of syngamy, forming a zygote and making the plant into again diploid condition. So this process roll over and over again, and in this way we call them the life cycle of heterosporous pteridophytes. This is all about the pteridophytes, how they reproduce, how they work, how the sporangia take place, and how their life cycle working. Still, you guys have any problem? You can ask me anytime. Thank you. That's all for